An electrical impulse can be transmitted through cells called neurons. By generating resting and action potentials, an electrical impulse can be generated and travel through the length of a neuron. These signals can then be passed on to another cell via chemical signals called neurotransmitters. The signal might pass from one neuron to another, or a neuron can pass a signal to some type of effector cell, such as a muscle, gland, or other target organs. In this video, we'll explore how a signal is transmitted through a neuron and then how that signal can pass between neurons. This is a neuron. A neuron is a specialized cell in the nervous system that transmits electrical signals. It consists of a cell body, which contains the nucleus and cytoplasm, and two main types of nerve fibers, dendrites and the axon. These are the dendrites. They are short, branched fibers that receive signals from other neurons. This is an axon. It is a single long fiber that carries impulses away from the cell body to the end of the axon, called the axon terminal. These fibers conduct electrical impulses, which involves rapid movements of ions across membranes that travel along the length of the neuron. The electrical impulse then arrives at the axon terminal. Here, the terminal forms a synapse or junction with other cells. Neurons have what we call a membrane potential. Membrane potential is the difference in electrical charge between the inside and outside of the neuron's membrane. We'll be referring to membrane potential as a voltage or a charge using the units millivolts or MV. When a neuron is not transmitting a signal, it is said to be at resting potential. Resting potential is a stable, negative charge inside the axon, typically around -70 millivolts. This resting potential is created by the sodium-potassium pump, a membrane protein that uses ATP to move 3 sodium ions out and 2 potassium ions in. Every cycle moves more positive charge out, 3+, than it brings in, 2+, contributing to a net loss of positive charge inside the cell. A sodium-potassium pump working through 3 cycles will move a total of 9 sodium ions out of the cell with a positive 9 charge, and 6 potassium ions in with a positive 6 charge. This creates a greater positive charge outside the cell than inside. This is happening all throughout the neuron from the dendrites to the cell body, axon, and axon terminal. The result is a concentration gradient with more positive ions accumulating outside the cell than inside. But how does this lead to an overall negative charge if only positive ions are moving in and out of the cell? The answer is there are many negative charged ions called anions that are trapped inside the cell. These negatively charged ions, in addition to the movement of more positively charged ions out of the cells, causes the overall charge inside the neuron to be negative. Because of this unequal charge distribution, the membrane is said to be polarized, or more negative inside than outside. And this is the resting membrane potential. When a neuron is stimulated, it generates an action potential, which is a rapid change in membrane potential. Now keep in mind we start with a neuron at resting potential, or a -70 millivolts. This is maintained by the sodium-potassium pump, which means we have more sodium outside the neuron and more potassium inside the neuron. However, when the neuron is stimulated, this changes. When a stimulus is strong enough, it will cause a change in the charge of the neuron, starting with the dendrites, which receive chemical signals from other neurons. If this change is large enough, it will trigger a series of events, which will occur section by section of the neuron, like a wave or a domino effect. So as the charge of the dendrite changes, voltage-gated sodium ion channels will open. Voltage-gated simply means when the charge is a certain level, the channels will open. In the case of most neurons, when the charge within the cell reaches about -55 millivolts, the voltage-gated sodium ion channels open. A charge of -55 millivolts is like a key that causes these channels to open. This allows sodium ions to rush into the neuron, depolarizing the membrane. Why do they rush in? Remember, the sodium-potassium pump has moved a lot of sodium out of the neuron, building up a concentration gradient. This local depolarization changes the neuron to a positive charge in this one section of the neuron. It then triggers the next segment of the neuron to do the same thing as it moves across the dendrites to the cell body and then finally to the axon. The axon then opens its sodium channels, and the impulse continues to propagate down the axon like a wave. Watch this animation a few times to get an idea of what propagation looks like. So following this action potential graph, at rest, we know the sodium-potassium pump is actively working to pump sodium ions out of the neuron and potassium ions into the neuron. A stimulus is received and causes a change in the voltage to about negative 55 millivolts. This change is enough to cause full depolarization of the neuron, which means voltage-gated sodium channels open and sodium comes flooding into the neuron. This causes the neuron to become even more positive, as the sodium ions carry a +1 charge. Now, once the impulse or signal has propagated or moved down the axon, the neuron needs to prepare itself to receive another signal. How can it do this? Well, it needs to return to the resting potential. In order to do this, two things happen. Once the action potential has occurred and the internal charge of the neuron is about positive 40 millivolts, this will trigger two events. First, the voltage-gated sodium channels will close. This will stop sodium from moving into the neuron. In addition, voltage-gated potassium channels will now open. What do you think will happen to the potassium? With all the potassium ions that were being actively pumped into the neuron, we have a concentration gradient, and potassium ions will move down the gradient, from inside the neuron to the outside. As this happens, the neuron is going through the process of repolarization. In addition, these sodium-potassium pumps will continue working to re-establish the resting state and continue pumping sodium out of the neuron and potassium into the neuron. Potassium channels close when the charge reaches about negative 70 millivolts, but they close slowly, so extra potassium escapes. This makes the inside even more negative than the resting potential, a state called hyperpolarization, or the refractory period. This is corrected once all the potassium channels close and the sodium-potassium pump works to restore the resting potential. Again, watch this updated animation a few times to get a sense of the entire process. How quickly all of this occurs can vary quite a bit. There are two key factors that influence the speed of nerve impulses. First, the axon diameter is important. Large diameters mean less resistance, so impulses travel faster. For example, giant axons in squid conduct impulses faster than smaller axons. The giant axon is a highly specialized, large axon that plays a crucial role in the squid's escape reflex and jet propulsion. In addition, myelination can change the speed of a nerve impulse. Myelination refers to a fatty insulating layer called the myelin sheath around the axons of certain neurons. This increases the speed of nerve impulse conduction. In myelinated axons, the impulse jumps between gaps called nodes of Rambier in a process known as saltatory conduction. This is much faster than in non-myelinated axons where the impulse travels continuously along the membrane. What this means is we only need sodium-potassium ions moving in and out of the neuron at the nodes instead of all along the axon, causing the impulse to jump from node to node. The presence of myelination can change the speed of impulse from 1 meter per second to 100 meters per second. Motor neurons that control skeletal muscle or voluntary movement are myelinated, as are neurons that control our reflexes, like the knee-jerk reflex. Some examples of neurons that are unmyelinated include interneurons in the brain and spinal cord, which involve signaling over very short distances. Neurons involved in involuntary control of organs, such as our heart or digestive system, and sensory neurons for pain, are also unmyelinated. Now that we understand how myelination and axon diameter affect impulse speed, let's talk about how we can use data analysis to describe and evaluate these relationships. When we examine relationships between two variables, like axon diameter and conduction velocity, we often describe them using the terms positive and negative correlation. A positive correlation means that as one variable increases, so does the other. In our case, as axon diameter increases, conduction velocity also increases. This is a positive correlation. A negative correlation, on the other hand, means that as one variable increases, the other decreases. For example, as the size of an animal increases, the conduction velocity decreases, showing a negative correlation between animal size and conduction velocity. To quantify how strong these relationships are, we use the correlation coefficient, also known as R. This value ranges from -1 to +1. A value of +1 indicates a perfect positive correlation. A value of -1 indicates a perfect negative correlation. A value near 0 means no correlation. You don't need to be able to calculate the correlation coefficient, R, on your own. You can use Google Sheets or Excel for that. But you do need to understand what it means. What does a correlation coefficient of negative 0.995 tell us about the correlation between conduction velocity in relation to body mass? Well, it tells us there is a negative correlation, and the value is close to -1. So this relationship is very strong. How about the correlation coefficient of conduction velocity versus axon diameter? A value of 0.999 indicates a positive correlation, and the value is also very close to +1, indicating there is a very strong correlation. We can go one step further. The coefficient of determination, or R squared, tells us how much of the variation in the dependent variable, in this case conduction speed, can be explained by the independent variable, such as axon diameter. For example, an R squared value of 0.999 means that 99% of the variation in conduction speed is explained by variation in axon diameter. That's a very strong relationship. The R squared value of conduction velocity versus body mass is 0.991. This also indicates a very strong relationship. 99% of the changes in the conduction velocity can be accounted for by change in the body mass of animals. As one additional example, we can view the relationship between the length of an axon and its conduction velocity. If we look at the scatter plot here, we can see that some shorter axons have a fast conduction velocity and some have slow conduction velocity. We see the same thing with the longer axons. They can both have fast and slow conduction velocity. When we add a trend line to the scatter plot and calculate the R and R squared values, R is equal to 0.110. This is very close to 0, indicating that there is a very weak correlation between axon length and conduction velocity. When we calculate R squared, the value is 0.012, indicating that less than 1% of the variation in conduction speed is explained by variation in axon length. There is a weak correlation, and we can say fairly confidently that conduction velocity is not impacted by axon length. So when you analyze real biological data, like how axon size affects nerve speed, look for both the correlation, coefficient, R, and coefficient of determination, R squared value, to evaluate how meaningful and reliable the relationship is. A synapse is a junction or site of transmission of a nerve impulse between two neurons, or between a neuron and an effector cell, like a muscle. The actual space between two cells is referred to as the synaptic cleft. Typical synaptic clefts are about 20 nanometers wide, or 0.000002 millimeters. We will focus on chemical synapses, where electrical signals that move across the neuron, as we discussed with depolarization, are converted into chemical signals which move between neurons, or a neuron to another effector cell. At a typical synapse, the signal passes in only one direction, from the presynaptic neuron to the postsynaptic cell. The presynaptic neuron is the cell that sends the signal. It contains synaptic vesicles filled with neurotransmitters. When an action potential reaches the presynaptic terminal, it triggers release of neurotransmitters into the synaptic cleft. The key role of the presynaptic neuron is to release the chemical signal. The postsynaptic cell is receiving the signal. It has receptors on its membrane that bind to the neurotransmitters. Binding of the neurotransmitter may trigger an electrical response by stimulating the cell or neuron and triggering an action potential or other response. Its key role is to receive and respond to the signal. Let's dig a little deeper into how neurotransmitters are released. When an action potential reaches the presynaptic terminal, the end of the neuron, it's going to cause a series of events to occur. First, the depolarization, or positive charge at the axon terminal, causes voltage-gated calcium channels to open. Calcium ions enter the neuron and act as internal signals. The calcium ions trigger vesicles filled with neurotransmitters to fuse with the membrane and release neurotransmitters into the synaptic cleft. This is a chemical signal being passed. For this reason, calcium ions, just like sodium and potassium ions, are essential for neuron function. How will the postsynaptic neuron respond? The neurotransmitters diffuse across the synaptic cleft and bind to the transmembrane receptors on the postsynaptic membrane. If the neurotransmitter is excitatory, like acetylcholine, it causes sodium channels to open, allowing positive sodium ions to enter the cell. The stimulus causes a small depolarization that brings the neuron closer to firing an action potential. If the stimulus persists and is strong enough, it will trigger the neuron to undergo an action potential. Acetylcholine is a common neurotransmitter seen in many types of synapses, including neuromuscular junctions, the synapse between a motor neuron and a muscle fiber, ultimately causing muscle contraction. After acetylcholine or other neurotransmitters transmit a signal across a synapse, they do not remain in the synaptic cleft. Otherwise, the postsynaptic cell would be continuously stimulated. Instead, several processes can occur to terminate the signal and allow the synapse to reset. Enzymes can break down the neurotransmitter. Acetylcholine, for example, is broken down by the enzyme acetylcholinesterase. This occurs in the synaptic cleft, producing acetate and choline. Choline is often reabsorbed by the presynaptic neuron to be recycled. Many neurotransmitters, like serotonin, dopamine, norepinephrine, are taken back up by the transporter proteins in the presynaptic membrane. They are either reused or broken down within the neuron. Lastly, some neurotransmitter molecules may diffuse out of the synaptic cleft into the surrounding extracellular fluid, lessening their effect on the postsynaptic neuron.